chapter 18. It's not very long. We're going to talk about neural mechanisms of learning memory. Okay, yeah, isn't that pretty? Research shows that memory formation can take place in the development of simple neuronal changes all the way to complex neural changes. Memory is only formed when neuronal changes take place. Simple neuronal circuits may receive input from more complex superordinate or modulatory circuits. So there has to be a neuronal change in order for memory to take place. This is why we are so brilliant. Are you ready for this? However, some memory requires more complex neural connections, relatively complex networks called cell assemblies. And of course, this is what a cell assembly looks like. This is one of the reasons why when you're trying to remember something, sometimes you can't find the right cue. In order to find the right cue, you've got to find this spot right here. But there it is. Okay. Maybe you've got information that is this spot, and you've got to make all these connections before you can get to this spot. That's how it works, and this is one of the reasons why older individuals might have a difficult time finding these things because they don't have the right cues. Many of the changes in neuronal structure can be measured physiologically. These changes may be presynaptic, postsynaptic, or both. Many researchers believe that long-term memory requires significant neuronal structural changes. Donald Hebb suggested that if a neuron frequently stimulated another neuron, that it would make a permanent change in the stimulated neuron. In other words, practice makes perfect. And what we have learned is that if you can see something or learn something 3 times, if you can go over material 3 times, then it is permanently in your memory, or it's in there for a while anyway. And then all you need to do is find the correct cue, and you can go back to that memory. A Hebbian synapse is a synapse that has changed because of frequent stimulation. This means that the synapse is not only active in stimulating other neurons in the area, but that it is frequently stimulated as well. Neurons that fire together wire together, and that's what we saw with the cell assembly. This frequent stimulation follows Hebb's dual trace hypothesis. Research actually done by the authors of this text, dealing with the weight of rats' brains and a standard environment, an impoverished environment, and an enriched environment, proved that rats who were raised in an enriched environment had heavier brains than rats raised in the other 2 environments. In other words, the ones in the enriched environments had much heavier brains than rats that were raised in a standard environment and an impoverished environment. And this may be one of the reasons why individuals that live in households where there is a lot of stimulation are more intelligent than individuals that live in a boring, dull environment. The area of the brain that had the most significant growth was the cerebral cortex, of course. That's the outer covering of your brain, but especially the occipital lobe. That's the part that you see with. That's so the more visual stimulation you get, like reading, interacting with the environment, the more intelligent the individual is going to be. The greater the synapses, the heavier the brain. Further research has proven that much of the change takes place because of dendritic branching. Dendritic morphology has been shown to be constantly changing. Fine filaments extend from the dendrites. These are called filopodia. If these filopodia make contact with an axon, they may form dendritic spines. And now we have a connection. Now we have one of these. There we go. There you go. All these neurons are connected. This is a cell assembly. And this is a memory. This is how it works. When a synapse receives repeated strong stimulation, it creates a stable and enduring increase in the effectiveness of the synapse. And this is termed long-term potentiation. This can especially be seen if the neuron experiences tetanus, a flurry of intense and constant electrical stimulation that creates a permanent change in the synapse. Remember, the more stimulation you get, the more likely things will be wired together. The neurons fire together, wire together. And this is known as tetanus. Tetanus is just overstimulation. The hippocampal formation is formed in two interlocking C-shaped regions, and these are the two C-shaped regions, as you can see. There's one of them, and there's the other. These are the hippocampus, the dentate gyrus, and the subiculum, the hippocampal gyrus. These three structures are divided into three regions, the CA1, CA2, and CA3 from bottom to top. NMDA glutamate receptors acts as an antagonist preventing long-term potentiation. But NMDA does not affect long-term potentiation that has already been established, so you can't lose memories. This is one of the reasons why you remember so much stuff. Because if you have created that long-term memory, then the long-term memory won't go away. That's why it's a long-term memory. AMPA glutamate receptors increase long-term potentiation. Activation of NMDA receptors leads to an increase in AMPA receptors. When glutamate is released in a synapse that has both AMPA receptors and NMDA receptors, the AMPA receptors require only moderate stimulation to be activated, whereas the NMDA receptors require a greater amount of stimulation. Remember, NMDA blocks long-term potentiation. AMPA accelerates long-term potentiation. This occurs because magnesium blocks the NMDA calcium channels, effectively keeping the NMDA receptors from firing and allowing you to remember things. This is so boring. You're increasing your NMDA. Wow, this is really exciting. You're increasing your AMPA, and you're more likely to remember something. If the AMPA receptors become stimulated enough, it depolarizes the membrane, removing the magnesium and allowing the NMDA to fire. This allows a large amount of calcium to enter the neuron. So the NMDA can only be activated when the cell is depolarized and there is glutamate present. The large influx of calcium activates protein kinases, enzymes that enables a phosphate group, PO4, to attach to a protein molecule. Phosphate is PO4. Protein kinase A, PKA, protein kinase C, PKC, calcium calmodulin kinase, CAMK, and tyrosine kinase, TK. Calcium calmodulin K affects AMPA in two ways. It phosphorylates AMPA receptors, increasing their conductance of sodium and potassium ions, making it more likely that you'll remember something. It promotes the movement of AMPA receptors from the interior of the neuron to the surface of the neuron, of the membrane. Calcium calmodulin K leads to the formation of intermediate term memory. Protein kinase C leads to the formation of long-term memory. Protein kinase A leads to long-term potentiation, which in turn helps to establish both long and intermediate term memories. These are enzymes that are helping you remember things. The activated protein kinases not only help to phosphorylize proteins, but they also trigger the synthesis of new proteins. The protein kinases activate CAMP responsive element binding proteins, or CREBs. CREB in turn binds responsive elements. The elements that CREB binds to are nucleotides. The nucleotides that are affected are in the promoter regions of DNA adjacent to genes. This changes the rate of transcription of adjacent genes. This in turn alters the growth and differentiation of the neuron. If the protein that recognizes Krebs is genetically disrupted, it causes a syndrome that is characterized by intellectual disability. So the more Krebs that you have, the more stimulation that you get, the more likely that you'll be an intellectual giant. If Krebs is genetically disrupted, then we have an intellectual disability. But there are also Krebs antagonists. Inducible CAMP early repressor, ICER, produces 4 proteins that block Krebs gene transcription. These proteins block Krebs by binding to the genes themselves, but do not promote transcription. If too many ICERs are present, Krebs will not induce long-term memory. Another Krebs antagonist is phosphatase 1, which inactivates Krebs. Now, why in the world would you not want to remember something? Why don't you just remember everything? And the answer is some memories are painful. Some memories need to be gotten rid of. Some things need not be remembered. So this is why we have the ability to remember a lot of things, and this is why we have the ability to block certain memories. It is thought that these Krebs antagonists exist to pair unwanted memories or to keep them from forming to begin with. And this is what trauma is all about. If we have a traumatic situation that we don't want to remember, this is why we have this mechanism to block our memory. The memory process takes place over hours. For the first hour, protein does not have to be synthesized to maintain the memory. It is just maintained. But for the memory to form long-term potentiation and hence long-term memory, Krebs has to be activated and protein synthesis has to take place. So how does this take, why would we do this? We're trying to put this in our permanent memory store. So that's when we try to activate the Krebs. After 3 hours, select proteins that have been synthesized will decrease while others must increase to maintain the long-term potentiation. At this point, the affected neuron will signal the presynaptic neuron to release more transmitter to maintain the signal. This is known as a retrograde signal, one from the past. Memory retrieval requires finding the proper cue to connect the individual with the neuronal bundle that contains the information. When the memory is found, research demonstrates that the structure of the memory becomes unstable and susceptible to disruption from protein synthesis inhibitors. If there are no changes, the memory will be reconsolidated and return to a stable status. This is, of course, necessary for memories dealing with a person's behavior, where inappropriate conduct must be repaired immediately. In other words, we remember good stuff and we forget bad stuff. Sounds like a Chumbawamba song. I get knocked down, but I get up again. Okay. Epinephrine affects memory formation by influencing the amygdala. It does this by forcing the release of norepinephrine in the amygdala. If the individual takes propranolol, the amygdala's memory-enhancing effects are blocked. And this is what they tried to do in Afghanistan and Iraq. They tried to reduce the amount of PTSD from combat situations by giving individuals propranolol. But one of the things that they discovered was that you couldn't make people not remember things. Propranolol does block the emotional part of it. But it couldn't block people thinking about situations over and over and over again. And that's how you put things in long-term memory. Opioid peptides will also block the release of norepinephrine in the amygdala. Since there are several major emotions, different neurotransmitters and structures come into play with the amygdala to create memory. Adrenergic neurotransmitters, opioid neurotransmitters, GABAergic neurotransmitters, and cholinergic neurotransmitters. The amygdala influences organs that are connected to it through neuronal connections, the hippocampus and the caudate nucleus. Propranolol works by taking the emotion out of the memory. The memory will still exist, but the details aren't as distinct. And we tried it, and it really didn't work. It didn't decrease the amount of PTSD that was coming out of the combat situations in Iraq and Afghanistan. The brain as a whole shows a loss of weight beginning at age 30. The frontal cortex loses more weight during this decline than any other portion of the brain. The hippocampus also shrinks with aging, being one of the reasons for the elderly to display memory problems. Another area of the brain that degenerates with age is the septal complex that provides input to the hippocampus. Since most of the neurons in the septal complex use acetylcholine as their transmitter, degeneration of the neurons or acetylcholine-producing cells lead to memory problems. Alzheimer's disease is such a memory problem. And that is the end of the chapter. So, had a good time this semester. I'll see you for the next class.